Okay, everyone, welcome again to the last talk of this session. Amitabh Varshney received his PhD in 1994. Fred Brooks was his advisor. He worked in computational methods to analyze the business end of proteins, the solid accessible surface, and worked with many people who did biochemistry and built on his visualization and his modeling techniques. He went to University of Maryland, where he's now the director of the UMIAC's University of Maryland Institute for Advanced Computer Science, I'm guessing, where he's doing fantastically, and he's come to talk to us about revealing patterns in the injured brain. Thank you.
Thank you very much, Ayan. It's such a pleasure to be back here. The last time I was here, I was defending my PhD dissertation in this very room. And nothing has changed. Jan, Fred, David and Jane, all of my friends, we are all, it's the same thing. And in some ways, I'm still learning from my teachers and friends just like I was at that time. It just feels so similar. So I wanted to give you a little bit of a background as to how this whole project started. So about 3 years ago, I'm a professor at the University of Maryland at College Park in computer science. And we don't have a medical school. And the University of Maryland at Baltimore is where the medical school is. So the president of our university got together with the president of the University at Baltimore, and they decided that it would be good for both campuses if the computer scientists at College Park were to work with the biologists, the medical health professionals at Baltimore. And this is where a degree from Carolina keeps giving. So they looked around, and there weren't that many people who were actively working at any point in their life on large interdisciplinary projects. So here I have to really go back and thank Dave and Jane Richardson for having so patiently worked with me throughout the PhD dissertation, teaching me a whole new vocabulary of working, what does it really mean to work in an interdisciplinary area. And so when the call went out to find people who could talk with folks who were not trained as computer scientists, I was one of the few people left standing. And so really made the most out of it and started working on this project. So the project is about understanding traumatic brain injury. And this is basically concussions. There are lots of causes for it. It's a significant challenge in the national economy. And one of the biggest problems is diagnosing mild cases of the traumatic brain injury. So the significant ones you can find out. The mild ones are the harder ones. So this is where you have a fall, you get up, you go to the doctor, they get an MRI done, and they say, well, everything is fine, go home. And you come back home, and 2 or 3 months later, you start having problems. You start forgetting things. You start having difficulty in reaching decisions. And so there's this progressive neurological degeneration that happens over time. And right now the thought is that if you could diagnose this early, there may be a chance for interventional therapy. But if you cannot, then of course you're losing that chance. Another big cause of this is football. And there was the big discussion about this throughout the country a few months ago, where the NFL denies that there is any issue, and yet the players believe that there is an issue where the way the sport has evolved, a lot of head is used in forcing their way through to the opposing team. So what is the technology out there? So one of the things that I think is very important and helpful, this is another thing that I learned here at UNC, is whenever you are working on a new area, it's always better to work in an area where there's a technology catalyst. So when I came here, the department had just acquired MassPower. And it was amazing to work on something that highly parallel with only, I don't know, was it 4 kilobytes of memory or lesser per node? But learning to program massively parallel processors later on became very, very important in programming the pixel planes, where it was very fantastic to work on the procedural textures project. And later on, GPUs, where Mark Harris was just talking a few minutes ago, and this whole, the same skill, you keep coming back to it in different settings and different contexts. So in this case, the driver was the diffusion kurtosis imaging. So everybody has heard about MRI, everybody has heard about the diffusion tensor imaging. Diffusion kurtosis imaging was something that was just getting started at Maryland, Baltimore. So it felt like this might be something interesting, and there might be an interesting advance and some interesting impact one could have on this field. So basically what this kurtosis imaging gives you is about 60 vectors which indicate the amount of water molecule motion at every point in a volume. And this water molecule motion is basically giving you the microstructure of the brain at that point. So the directions in which the molecules can move more, that's where the space is. The direction where they cannot move much, that's where the brain matter is. So here is a vector field with just one vector per point. And you can draw all these vectors, and of course it's not quite as, you get some sense as to how the local flow is happening, but you don't get a very good sense. So one of the things that visualization guys got inspired by was looking at the astronomy images where the telescope looks at a given direction, the shutter is opened, the telescope does not move with the stars, and you get this kind of a flow pattern where the stars are moving, and you have a very clear sense as to how the flow is. So this similar idea was adapted by Cabral and Leadham in SIGGRAPH 93, and essentially what they did is they took the starting point of the vector field, they continued in that direction, interpolated further, but then they convolved this with a random dot pattern, similar to what happens in the case of stars. And you get this whole line integral convolution, which gives you a really nice detailed view of what happens when you're looking at these vector fields. So this was the starting point, but the problem is it's only still one vector per point, and now we have 60 vectors per point. So how do you now visualize this? So before we get to 60, let's talk about 6 vectors per point. So this is the diffusion tensor imaging, and a lot of the early work in this area has basically used the idea of using ellipsoids. So ellipsoids are representing your 6 vectors per point and approximating it. The colors are just showing you what direction the ellipsoids are pointing, left to right, top to bottom, front to back. But essentially the idea is to use ellipsoids to give you a sense as to how the local vectors are moving. And that's still very good, but if you go more, it becomes very hard. So the nice part about this diffusion kurtosis imaging, which is what was so exciting about this, is that it can detect changes in the microstructure not only in the white matter tracts, which are the coarse highways of the brain, but it can also detect changes in the gray matter, which is the much more finer, subtler microstructure of the brain. So when I started talking with the radiologists on this, they said, oh, the DKI measures the 4th order diffusion tensor. And the thing was, well, what the heck is a 4th order diffusion tensor? So you start looking at it and they say, oh, it's the kurtosis, it's a kurtosis. Well, what is kurtosis? So the best way I could figure out to really get my head around this was the first order is what would be the equivalent of a sphere. The second order would be the ellipse or the ellipsoid, which is measuring the standard deviation. And the third order would be the egg, which has a different, it is pointed in one direction more than the other, and this would be the measure of the skewness. And much as I tried, I couldn't find a really good example of kurtosis. They kept, oh, it's the deviation from the skewness. So the best thing I could find was this. And this, of course, tells you the age-old question of who came first. So now if you take this whole idea and bring it back, it's, you can use glyphs. It's very hard to make sense out of these. This is what these glyphs, which would be the counterpart of using the ellipsoids. If you extend it up one level higher, they look like this. It's very hard to make a sense of. There's occlusion problems, there's orientation problems, their density, lighting, they're very expensive to render. So none of this was working. And so at this point, and if there are any PhD students in the room right now, I have to tell you this. All of us, professors and researchers, we always tell you what worked. We don't tell you how hard it was to get it to work. So don't lose heart. I mean, this is a regular process. It took us 1.5 years. And so this is the point. And after this point, for 1.5 years, we kept trying again and again and again. We would come out with some visualization, go to the radiologist, say, does it work? Does it make sense? They would say, what is this? I mean, this doesn't make any sense. Then you come back and you try again, and none of that is making it to the talk, because the whole point of the talk is to show you what worked. But there's a lot that doesn't work, so don't lose heart, keep at it. Eventually, hopefully, things might work out. And so coming back to this question of how does one visualize a field with 30 vectors per point, let's take a slight segue. There was this early work on lighting. So if you look at a movie like Ender's Game, you look at the real actors, you look at the computer-generated imagery next to them, and one of the things you notice is that the lighting is made consistent between the actors where the light is coming from this direction, as well as with these virtual objects where the light is coming from that direction too. And so light consistency is often considered very important in movie making. And in some ways you could argue that it would make sense, because we have all grown up on a planet with a single sun, and therefore lighting and everything around us is physically based. So lighting consistency would be considered very important to our natural senses of vision. So in 2006, these three researchers, Ostrowski, Sinha, and Kavanaugh, did this research on finding how hard is it for people to detect illumination inconsistencies in carefully photoshopped images. And it turns out it is very hard. So in particular, they tried these 9 cubes, randomly oriented. One of them is inconsistently lit with respect to the others, and asked people, can you find the one cube that is lit inconsistently with respect to the others? And it was very hard for most people to find this. Can you find one? Well, this is a smart group. So in general, it took users 8 seconds to find it, and then they were wrong 30% of the time. So it's this one that is lit inconsistently, right? So artists have been using this inconsistent lighting for several hundreds of years. This is the George Washington crossing the Delaware, way back when Delaware River had ice on it. And one of the things you notice on this is everybody is being lit from the left, except for this guy who's being lit from the right. And one of the reasons art historians who look at this stuff suggest is the reason is because the artist wanted to convey this subliminal message of the effulgence of George Washington radiating everybody with his brilliance around him. Same thing is true in this Jan Vermeer's painting, which is on the cover of the Foley Van Damme Finer Hughes book. And you look at it and you say, well, everything looks great. And you ask people, what is inconsistent? And they look at it, and after a while, the most common inconsistency people find is that the girl is looking straight down at the piano, whereas over here she is looking at the left. And so there is this is a mirror. So there's a geometric inconsistency you can find, but the lighting inconsistencies are very hard to find. And so if the sun is up over there casting a shadow like this, and that shadow is consistent here, why is there not a shadow over here? And here again, the art historians say, well, the reason is you would like the focus to be on the girl who's the centerpiece of this painting, and not having a bright, shadow go past the foreground would distract from having the attention go to the centerpiece. So this idea has been used before in visualization. In 2003, Akers and Manish Agarwal and others at Visualization presented this idea of, if you were to light a skull with one light source, that's how the skull would look. But the scientific illustrators do not do that. They use several light sources, and they light things up from different directions. So we thought this was a great idea, and we did a follow-up paper on this, where we were using local inconsistently lit objects to light things up so that they would reveal more of their structure than otherwise possible. And after I published this, so this was published with my students, we published it at IEEE Visualization, I was so excited and proud. I met Tony DeRose from Pixar and said, Tony, hey, we did this really cool work where we are lighting objects inconsistently. And we published it at IEEE Visualization. And he said, so what's new about it? We have done this for years at Pixar. So that's another thing to keep in mind, is just because something has not been published doesn't mean it's necessarily new. It's just that people haven't even bothered to publish it. So that was very humbling. So another area, so we will be using inconsistent lighting in a moment. Just keep it on the side of your of your table for a moment. Another area which was really big and really important is this whole area of spherical harmonic lighting. And the idea here is, instead of using point light sources where you can light an object from one point, you use the entire hemisphere to do the lighting. So light comes either from all directions or only from above and only from below. And this idea was first used, although not for lighting, but for representing diffused reflections by Basri and Jacobs, as well as Ramamurthy and Hanrahan in SIGGRAPH, and Basri and Jacobs in CVPR. And the idea was you could use this kind of lighting to give you a much better idea of the surface structures very cheaply at runtime compared to the point light sources. And here are all the basis functions on the Triceratops. Well, no, not what is this model called? It's not the Triceratops. It's the game, huh? Armadillo Man. That's right, Armadillo Man. That's it. Yeah. And this is being used a lot in games these days. So this is the normal lighting. This is the equivalent of spherical harmonics lighting. It's from the game Fable Legends. And basically what it gives you is, you can see it is able to reveal the underlying structure much better than otherwise. So we decided to use both of them together. We said, let's assume that the lighting is not consistent, and let's use spherical harmonic lighting just because it's so efficient and reveals the underlying subtle patterns much better. So this is what we have done here. And so here are your actual Kurtosis tensors. These are basis functions of the Kurtosis tensor. These are all the 60 vectors sitting on a voxel being approximated in these ways, and here are all the lighting functions, and you compute their product. So this times this is this color, this times this is this color, and you use different lights and different tensors and just do dot products, and they are very efficient to do. And so you can do spherical lighting, you can do directional lighting, you can do radial. This is what happens when you have 2 lights, this is what happens when you have 3 lights. So the motivation for this kind of work is really, if you look at velvet, which has very fine grained structure, and you light it, small subtle variations of the velvet are captured. In some ways the brain microstructure is similar, and by lighting it you can bring out that subtle structure of the brain. And so this is changing the light in different directions. And this is showing you the red, green, and blue lights coming in from different directions. And as they change, you can see how the structure changes. So this was very exciting. We could finally see some difference in the structure. So we went back to our radiology colleagues and showed them this. And they said, yeah, I mean, it's interesting, but does it really tell us what we are looking for? And so at this point we said, look, okay, this is getting very hard, so you have to give us some brains. And they say, yeah, sure, here is, they gave us 30 brains, 15 normal, 15 with various kinds of concussions. And so we started trying these out. And so here is, so the lighting helped, but what really helped was not the lighting per se, but to take the total amount of light intensity and use that to map into opacity. So basically, this is a normal subject. This is the patient with a traumatic brain injury. And what you can do is by changing the light direction, you can highlight these different regions of the brain with or without the traumatic brain injury. And this really does give you so much more insight into where are these regions where the subtle texture, the subtle fabric of the brain has been disrupted. And we did the same thing. This is the global orientation. By global orientation, I mean consistent lighting throughout. This is the local orientation. So what we did is we said, let's orient the light locally aligned with the local kurtosis tensor, the dominant tensor of the local kurtosis tensor. And it gives you a lot more information than if you just did it globally. So this whole idea of inconsistent lighting locally together with spherical harmonic lighting allows you to see much more of the brain than otherwise possible. So the left three, mean diffusion, fractional anisotropy, and the principal diffusion direction, this is the state of the art. And the right two are the ones that our approach shows. And so here is the traumatic brain injury over here, the white regions, and here is, this is being shown in 2D, this is being shown in 3D, and you can see things much more clearly than otherwise. So another case study they showed us was the visualization of this mild brain injury where the MRI was taken 10 days and 6 months after the injury. And the brain had atrophied a lot in this case, and it's very clear using this approach how much the brain atrophy has been. This is another visualization where there's some frontal lobe damage where the recovery is not that visible using the traditional technique of fractional anisotropy. Both of these brains look very similar, but using this idea of spherical harmonic lighting with locally oriented, local orientation, you can start to see that the recovery is much more clearer here than over here. The tumor lights up very easily and very clearly in our case than in the previous cases. And so again, these are all cases where very subtle changes in the brain were amplified and shown very clearly with this approach. So here is a testimonial from the
This is what Sujal's work is extremely important for us because it allows us to visualize these.
Oh, by the way, that is the PhD student who did all the work and commuting back and forth from College Park to Baltimore and back.
Microscopic or microstructural changes in the gray and white matter at the same time, and also be very close to the basic science where you're doing microscopy. So this opens up a window for us to visualize microscopic processes in the brain that we were otherwise unable to do in the past.
So as compared to diffusion tensor imaging, diffusion kurtosis imaging deals with a much more complex tensor structure, the kurtosis tensor. Currently, there's not yet a very good way of visualizing that. Sujal's work is a breakthrough that finally provide a way to visualize the kurtosis tensor in a very useful way and then very intuitive manner that will help the radiologist to find abnormal areas and then to understand what's really going on in the tissue microstructure.
This is what Sujal.
So to conclude, I mean, the basic idea here was to see what all machinery we could take from computer graphics and visualization and bring it to bear on this problem of looking at large radiological data sets and make or reveal what was in these in a way that would make sense to the radiologists. So one of the things that is really exciting right now is I think we are seeing a huge resurgence of interest. The headsets are becoming cheap. This Christmas, everybody's going to get a headset, whether it's from Microsoft or Facebook Oculus or HTC Valve, remains to be seen, but there would be a lot of these. And projectors, imaging sensors, GPU processing is really reaching a pinnacle almost. And so what would be really exciting is to see how we can co-develop a lot of these technologies and the implications for a whole set of applications in the life sciences, consumer applications, as well as science, medicine, and engineering. So I would like to very warmly acknowledge all the work and help and support and mentoring that I have received here at UNC Chapel Hill all these years. It really is a family. And one of the things that I think characterizes this family is this constant giving and forgiving that happens. And I, someone like me fits very nicely into this family because I believe in getting and forgetting. So it's a perfect combination. So thank you very much, guys. Yeah, Rich.
So did you look at running PCA on these 60 vectors to see if some axes are more important and maybe you can reduce the space?
Yeah. So actually that whole diffusion tensor imaging is essentially computing PCA. And so if you were to take these Kurtosis vectors and run PCA, the results are very similar to what you get from diffusion tensor imaging. And so it's very hard to, so basically you end up losing a lot more of the finer structure. And so that's why, so that's one of the things I didn't talk about. So we used PCA, it didn't work, so then we went with these funny looking glyphs, and these glyphs were too much geometry. And so that's where went to the lighting. Oh yeah, thanks, sorry. So the question was, why not use PCA? And the answer was, yeah, PCA works somewhat, but it's also overwhelming to the users. Dinesh and Jack. Dinesh, yeah.
Great talk. You mentioned about, I'm trying to understand the first time I learned about kurtosis. It's the fourth order you explained very well. Is something intrinsic about fourth order giving you more information than what mean and this? Because here a PCA is also a first order method,
Yeah.
So I'm just trying to geometrically think about there's some inherent information which is coming from this fourth order which the first and second order could not capture.
Yeah. So this is, so there is basic, so what is happening is, I guess okay, now I see the question. So the question is, why the fourth order kurtosis? Why not first, second, or third orders? And I think this is, goes to the heart of the problem. The problem is we are trying to diagnose mild cases of the traumatic brain injuries. So because they are mild, they don't get revealed as well in the first, second, and third orders, but they show up in the fourth order. So these are very subtle changes in the brain microstructure, and they're subtle enough to not get revealed early on. And this is one of the challenges too, because as you go into the higher orders, noise becomes more in these. And I remember this from the paper you mentored me on, Dinesh, here, on this higher order and the robustness. And this was really, I mean, it's very high. So the more high order you go, the more fragile this whole thing becomes. And this is where this new technique comes in, because to try and make up for this loss of the signal. That's the reason why you have to take 60 measurements per voxel in these directions to try and have some semblance of order. But that's really the heart of this whole problem. Jack,
so
yeah.
My question was sort of the flip side of that is, you were showing us the difference between the normal and injured brain, but you weren't showing us the controls. Have you done extensive studies of the same brain, different over the time periods and what they look like?
So no, this one, so the left is the normal, the right one is the traumatic. So one of the things, and this is basically where the challenge comes. So right now, we have done this for about 10, so we got this data set of 30. We are starting to work on 5 and 5, 5 normal, 5 with the mild traumatic. What we have to do is to do it on a much larger data set before something like this would be used in clinical practice. Now, one of the things which is sort of interesting is, and this I wouldn't have guessed, the traumatic injury patients are much more alike than the normal patients. And the normal patients are all over the map in some sense. So the diversity is more amongst the normals than with the injured, which kind of makes sense because they're. Another challenge that is facing our partners in this is it's very hard to find normal people to get their diffusion kurtosis imaging done. You have to end up paying them. The other group actually pays you. So that's another difference. But that's where this would lead to next, is to do large scale analysis over lots of brains to see, I mean, this was sort of the precursor to that, to see whether this signal persists in larger populations.
And I had a second question, which was, do you always have to wear a tie when you're collaborating with a brain surgeon, or is that only for the video?
No, they don't take you seriously otherwise.
It's kind of, almost similar to what Jack was asking. I'm just looking at your images. I don't know the theory behind it, but is it perhaps maybe in a normal subject, the red or the purple kind of moves around a lot more, whereas with the traumatic brain injury, it kind of stays in the same place over time. I don't know.
So what was, sorry, what the question is whether the purple.
See, even the normal subject has some purple, right?
Yes.
Indicating some amount of whatever that is equal.
Right.
I'm just asking whether that just moves around within the brain in a normal subject over time, as opposed to with
Right. So okay.
traumatic brain injury doesn't.
Okay, so that's so the question is, does the purple region, by the way, this is indicating high kurtosis regions in the brain, and does it move around a lot? So one of the things I didn't explain, I should have started with this, is that this is looking at the hardware of the brain. So this is not a functional MRI that is showing you the thought patterns or the metabolism in the brain as the brain thinks. So this is looking more at the hardware of how the brain microstructure is. So it would not change that fast. It's sort of very static. There is another version of this, which is we are working on right now, which is to look at how do the brain networks, there's a attention network, there's a default mode network, there's a saliency network, how do these networks get disrupted under mild cases of traumatic brain injury? So as an example, if you attach an eye tracker to somebody with a mild TBI, their eyes wander all over the whatever visual stimulus you present them. It's very hard for them to fixate. And so relating that to the brain activity is one of the things we're actually working on right now. But this is the hardware part. Yeah.
Thank you.
All right, well, thank you very much.